Okay, hi, welcome to Friday lecture. Happy Friday to you. Um, let's continue talking about allergies and the role of our immune system in allergic responses. Just quickly click on something here, make sure we're set up well. All right, good. We have a sort of a concept or a model of allergies that we call the hygiene hypothesis. And that states that because in first world developed countries, as children, we are sort of taught or exposed to constantly practice hygienic sort of practices, right, excessively, like washing hands with soap, always bathing every single day. And we tend to also be in environments that are more sanitized. This includes both our living environments that surround us inside of our homes, as well as things like plumbing, right, and sanitation. The hypothesis is that as kids, when you are first growing and developing your immune system, because remember, we are not born with a fully intact, ready to go immune system, especially our adaptive immune side, requires time, right, throughout childhood to be able to develop. That if we are depriving our immune system of being exposed to things that evolutionarily Mother Nature designed us to be sort of expecting to be exposed to, then perhaps our immune system doesn't really develop, right, with the same sort of proper awareness of what are threats and what are not threats. I mean, think about this, right? We wash our hands. We teach our kids to wash our hands a lot with soap. We bathe a lot. The types of environments that we live in are much cleaner, right? We have access to things like antibiotics. When you're a little kid and you get sick, you take antibiotics, right? All of these things may actually, in some way, slow or affect the normal natural development of our immune system. I mentioned parasites. I think none of you are surprised that if you go out into the forest, right, and you trap an animal, right, maybe you're studying that animal as a biologist, or you're fishing, catch a fish, right, you're not surprised if wild animals carry with them some sort of normal level of parasitic infection, right? I mean, even our pet dog, pet cat at home, they sometimes get parasites, right? And it doesn't really, I mean, it kind of maybe annoys us because we have to deal with it, right? But it doesn't really kind of shock us, right? Because after all, we think, oh yeah, they're animals, right? Sometimes they get worms or something like that. But consider this, right? Aren't we just animals as well, right? Aren't we evolved in a natural environment that would be exposed to such parasites as well? Over eons of evolution, wouldn't it make sense that our immune systems come ready to respond to certain levels of microbial exposures, parasitic exposures? And also, doesn't it make sense that Mother Nature would have expected that our exposure to such things would be an important part of the training of our immune system as it develops when we are kids? Our immune system, as we've learned before, right, has to have a certain calibration. In other words, it has to know when to actually take something seriously, right, and activate, versus when something is okay to just let go and not worry about. Right? This idea of calibrating then must come from as our immune system is developing when we were kids, based upon exposures, right? So it can properly sort of adjust thresholds above which the immune system reacts, below which the immune system is fine with. So excessive hygiene, right, eliminates our exposure to many types of pathogenic organisms, as well as exposure to other people and exposure to animals. Right? Vaccinations. We take vaccines to prevent us from having to actually experience the real illness. Right? Now, I want to be careful about this because I'm not trying to say that vaccinations are bad because somehow there's a chance that by being vaccinated and you don't actually experience some sort of terrible contagious disease, that, oh, your immune system is somehow losing out on the opportunity to learn from that. Because, right, the whole point is, right, it's a balancing act. Because if you actually experience that awful contagious disease, right, you have to fight for your life and you might not survive, right? So obviously, it's more beneficial if you can be vaccinated and not have to be exposed to that thing in the first place and have to fight for your survival and count on your immune system to save you, right? But it's quite possible, though, because we're vaccinated, we're not exposed to certain types of otherwise normal viruses or other illnesses that are transmitted through the population. I mentioned, my story about chickenpox, right? And how as a kid, I was a kid before we had chickenpox vaccines. I think the chickenpox vaccine came out in the early 90s, right? And I told you the story about how when a kid in the neighborhood would get chickenpox, if it was a good time of the year to get chickenpox, like the summer when there's not going to be any school to be missed, the parents would just get all the neighborhood kids together so that they could all spread chickenpox, right? And end up suffering from chickenpox for like a week and a half of blisters and itchiness and whatever, but at least you didn't miss school. And then after you're done with it, right, you're immune, right? So think about this, right? That exposure might have actually been part of what Mother Nature intended in order to properly develop a nice, strong immune system that could know, right, how sensitive it should be to worrying about different types of pathogens. So vaccinations, like being vaccinated against chickenpox, maybe takes a little piece of that opportunity for the immune system to properly calibrate away. Right? And over-reliance on antibiotics. Again, right, Mother Nature did not intend us to take antibiotics, right? If we're getting infected by a bacterial infection, Mother Nature intended our immune system to figure it out, right? And figure out how to save our lives, and in the process become stronger and better at doing it. So antibiotics that shorten the length of an infection, right, from maybe 2 weeks down to maybe 3 days, maybe that's actually depriving a developing immune system, right, the exercise that it needs to learn how to properly deal with things. So when you take all of this into consideration, right, that's why you see the hygiene hypothesis says that in first world developing countries where we have access to all of these different types of things, right, maybe what we are doing, right, is creating weaker immune systems, or at least immune systems that are not properly calibrated. Right? And if they're not properly calibrated, what happens? Right. So the development of the immune system might be held back. That's part of this hygiene hypothesis. It becomes poorly educated, poorly exercised, and inexperienced in fighting real world infections. And instead, because the calibration of the immune system, right, the immune system no longer properly is exposed to all different types of challenges. So it becomes more and more sensitive, as though the developing immune system is saying, wait a minute, I was designed to detect a whole bunch of different threats. I don't see those threats anymore. So therefore, maybe there's something wrong with how sensitive we are, right? So the immune system becomes more and more sensitive until it can start picking up on different antigens to get excited about, right? And as a result, once it gets so sensitive, those antigens that it begins to pick up on are not true threats, right? They are the antigens that represent allergens, things like the flower pollen, things like the pet dander or peanut butter, right? So that's the hygiene hypothesis. And I think it makes a lot of sense. And it's an interesting way of thinking about how our modern lifestyles are influencing, right, or impacting the way that our immune systems, after hundreds of millions of years, are designed to expect, or what our immune system is expected to encounter. All right, so let's review what we discussed last time when we first started talking about allergies. We said that all allergies can be categorized as one of 4 hypersensitivity types. We said that type 1 are the immediate hypersensitivities, right? So this involves IgEs that the body has previously made because the body was exposed to something that it mistakenly thought was a threat, like exposure to pollen or being stung by a bee, and the adaptive system made a bad judgment call, right? Because really the pollen or the bee sting, yeah, right? The pollen certainly is not going to pose any threat to you. Bee sting, yeah, right? I mean, it is painful, right? But it's not like it's designed to cause some long-lasting damage to you, the person who's stung, right? But your immune system might make a mistake and actually think, ooh, these are new antigens that we are going to select immunoglobulins and T cell receptors to be able to recognize and use as part of our memory, right? And so you would produce a lot of IgEs that then are coated on the outside of mast cells, and those mast cells line your mucosal layer. So unfortunately, the next time you're exposed to that pollen or the next time you're stung by a bee, right, all of these mast cells that have that memory IgE as part of them, right, release their cargo in their vesicles, and that causes strong physiological changes, right, like the tightening of the air passage, for example, as the smooth muscles around the respiratory tract contract, right? And also possibly anaphylactic shock as the blood vessels throughout the body, right? Some of the walls of the blood vessels of capillaries become more dilated and the blood pressure drops dramatically, right? So what I've done on this slide here is I've highlighted in colors the different cell types that we've discussed so far in the semester and tied them with the particular hypersensitivity type that we're discussing. So the pink ones, type 1, right, those involve plasma cells that are releasing IgEs, and then the mast cells that suit up with these IgEs. Right? Type 2, remember, hypersensitivity is also mediated by antibodies, but in this case, we're talking about IgGs. So in this case, you can see IgG production would involve B cells that are being selected for, plasma cells as well, and it would involve macrophages that then begin to target the normal healthy cells of the body if those normal healthy cells are presenting something funny, right? Because proteins in those cells might be modified by certain types of drugs, like penicillin, right? Inadvertently causing a mistaken sort of identity to happen. Type 3 are when you have immunoglobulins or antibodies cross-reacting with other antibodies, forming big complexes that your body has a hard time clearing or resolving. So again, these involve things like neutrophils. Involve B cells and plasma cells. They also involve neutrophils, right? Because neutrophils are part of the response that your body has to try to clear away many of these immune complexes. Neutrophils are attracted to these immune complexes, and unfortunately, the neutrophils can also cause damage to surrounding tissues where these immune complexes are kind of stuck in your tissues. And then type 4 was the one type out of the 4 that does not involve antibodies, right? We said that type 4 hypersensitivities involve T cells that are selected for to react against sort of modified peptides or proteins that look a little funny in the cells of your body because perhaps these proteins have been altered in such a way, such as from cheap jewelry like nickel ions, metal ions, or things like touching poison ivy, right? Causing your skin cells to present funny looking proteins. And then these cytotoxic as well as helper T cells start trying to mobilize an attack on your healthy tissues. All right, so let's take a look at some other examples of those granulocytes that might be involved in an immune response. Right? So mast cells are another category of these granulocytes. They contain a lot of histamine proteins. So mast cells are located in your tissues, such as the mucosal layer and the epithelium. Right? You don't find mast cells in the blood. The purpose of mast cells are they're meant to detect that there's an inflammation or an infection going on. So remember, they get suited up with IgEs. And so when they are triggered and release the contents of their granules or their vesicles, these types of compounds that are released stimulate inflammation, right? And they're also meant to help remodel or repair any tissue damage. So each mast cell has anywhere from 50 to 200 of these sort of vesicles containing different types of proteases and different types of cytokines. So a couple of examples of the contents of these are, for example, histamine, which we'll talk about. Histamine is a strong inflammatory cytokine, and heparin. So heparin is an anticoagulant, so it's a blood thinner. There are two main mast cell populations in your body. Of course, many of them are in your mucosal layers, right? That's where they do a lot of their job. And they can release certain types of protease enzymes like tryptases. So microbes might be digested by these proteases that are released by the mast cell. Other ones are in your connective tissues. So connective tissues are the tissues that kind of form things like muscle, bone, and the deeper layers of your skin. Right? So these mast cells might release chymotryptase to help remodel the tissue, right? So that damaged tissue can be repaired. So this cartoon on the bottom illustrates a mast cell that's being triggered to release its vesicles. And I think all of you now, by now in the class, realize that this cartoon, even though conceptually it gets the point across that it has these IgEs on its surface, and they're binding in this case to something like pollen, right? I think you all realize that this illustration is terribly drawn in terms of not being properly in scale, right? So for example, the actual IgEs on the surface of the mast cell, they should show that these IgEs are actually being held there on the surface by the mast cell by like Ig epsilon receptors or Fc epsilon receptors, right? So these IgEs are not simply sprouting out of the mast cell. They came as antibodies that are floating by, and the mast cell holds onto them. Number two, the size of the antibody, of course, is drawn completely out of scale, right? Because if you think about it, the antibody itself is a reasonably moderate-sized protein, right? And if in the illustration, these little purple dots that are coming out of vesicles of the mast cell are meant to be little cytokine proteins, right? Certainly the immunoglobulin shouldn't be this big. And of course, lastly, the pollen granule itself, right? The pollen granule might very well be much larger than a single mast cell is, many times the size, right? So the actual epitope on the surface of a pollen granule that's being picked up by the IgE, right, would just be a tiny little dot on here somewhere. So why do I show this to you? I show this to you because now that you guys have a stronger foundation of understanding what these different components are of your immune system, antibodies, cells, things like that, when you look at illustrations like this that are commonly sort of shown to the general public, right? Because these illustrations that are often distributed on websites to educate the general public about how things work, they don't care about being technically accurate, right? They just want to get the point across, almost like to get the public thinking about something, right? But now you know you can be careful and you can point out when you see something like this, what's wrong with it. Okay. Histamine is one of the primary cytokines, well, not cytokines, I'm sorry, is one of the primary molecules released by mast cells that trigger inflammation. Right? So histamine is actually derived from the amino acid histidine. So in this illustration here, this is the amino acid histidine. Right? So for those of you who took Bio 275, hopefully this doesn't give you bad flashbacks. But remember, it's an amino acid. So you have a carbon here, the central carbon that has an amino group on one side. It has a carboxyl group on the other side. And then it has some sort of variable R group side chain, right? Depending upon which of the 20 different side chains it could be, that defines what amino acid you're looking at. So this is the side chain of histidine. Right? So in the body, histidine is modified into histamine. So you can see here, it's very obvious that histamine comes from the amino acid histidine. Right? So I think all of you who suffer from allergies are familiar with at least histamine as being something that seems to be a big trigger of allergic reactions, right? Because when histamine is released by mast cells, it's received by histamine H1 receptor proteins on the surfaces of cells like smooth muscle cells and the endothelial cells that make the lining of your blood vessels. And this can cause inflammation. It can cause tightening of the smooth muscle. Those are the things associated with things like you being suddenly having itchy throat, itchy eyes, coughing, sneezing, difficulty breathing, right, as your airway restricts a little bit. Right? So histamine is also involved, though, in another important physiological human function that has nothing to do with allergies, and that is histamine regulates our sleep cycle. So the H1 receptors that receive histamine in the brain can be used, the histamine signals that are normally released and circulating around in the body can tell the body whether you should be alert and awake or if you should be drowsy, right, and go to sleep. So certain drugs, certain medications that are designed to be taken in response to allergies are called antihistamines, right? You know that, antihistamines. Things like common allergy medications that you take, if you start getting like sneezing a lot, itchy eyes, itchy throat, you take a pill, right? So how do these work? So antihistamine drugs like diphenhydramine, which is shown down here, right, they block the H1 receptors of cells and prevent them from being able to receive the histamine signal. So diphenhydramine, which is the component of Benadryl. Yeah, you all know Benadryl, right? Benadryl is diphenhydramine. When you look at this, you can see that there's a resemblance between diphenhydramine and histamine, right? This part here, there's a ring, and then there's like a little chain of carbons, and then in this case, you have a nitrogen, and you have a couple of methyl groups sticking off of it. But it also has this other ring here. Right? What happens is that diphenhydramine is just the right shape close enough that it can actually bind to the H1 receptor, just like histamine itself would bind to the H1 receptor. But because diphenhydramine, right, is a bit bulkier and has that extra sort of structure to it, it prevents the H1 receptor from actually activating, right? And so if it's bound to the H1 receptor, right, not only does it prevent the H1 receptor from activating things like your smooth muscle and your endothelium, but it also prevents histamine from being able to bind to the H1 receptor because it's in the way. Right? So the early generation of drugs like Benadryl, as they're flowing through your body because you take a pill and they get into your brain, they also prevent the H1 receptors in your brain from receiving histamine. So as a result, without receiving the normal histamine signal, right, your normal physiological sleep-wake cycle gets kind of messed up and you become very drowsy. That's why many antihistamines, especially early generation ones that were designed in, like, let's say the 70s, 1970s, right, they can cause drowsiness. Second and later generation antihistamines, things like Zyrtec, which is cetirizine, or Allegra, which is fexofenadine, right, they are tweaked in their structure so that they don't actually cross from the blood into the brain tissues as easily. And as a result, you still benefit from the antihistamine responses in places like your respiratory tract, right? So if you're having a hay fever attack or whatever, right, you can take Zyrtec or Allegra, but they don't make you drowsy because it doesn't mess with the normal histamine signaling that happens in your brain to keep you awake. Okay, let's talk about eosinophils. So eosinophils are another one of these granulocytes, and they're found in tissues that underlie the respiratory tract, GI tract, your genital tract, etc. They stain pinkish with the acidic dye eosin. That's how they're identified. And they, again, can release these highly sort of damaging types of compounds, molecules that they're carrying around. So eosinophils are usually very rare. But here's an example of an eosinophil floating through the blood, surrounded by normal, happy, healthy red blood cells, right, that are kind of mingling with it. Everybody's happy, everybody's good. But if this eosinophil is triggered to release its granules, so here you have some eosinophils on the right. See, these still have their pinkish granules in them, but these eosinophils in the middle have released the contents of their granules. Look at what happens to the surrounding red blood cells, right? A lot of damage, right, to surrounding normal tissues. Okay. Basophils are the rarest of the circulating leukocytes, and they can move between blood and tissue. They stain with basic pH stains such as hematoxylin, right? That's why they're called basophils. So we don't really know that much about basophil function. They are the rarest, but it seems that basophils and eosinophils, the production by your bone marrow of these two types of cells is reciprocal, right? You make more of one and less of the other. And we think, by and large, it has the same or similar functions to those eosinophils. So remember, allergies come from your adaptive memory. So most type 1 allergens that trigger immediate and potentially strong allergic reactions, most of these allergens themselves are proteins. Right? So when you think about the antigens that are stimulating them, what types of molecules stimulate these types of responses? The majority of them are proteins. Why? Because remember, proteins are highly variable in shape, but oftentimes proteins can be dried, they can be airborne, they can enter the respiratory tract. So let's look at this story here. So here's this guy, he's maybe moving out to the country. He's lived in the big city, all his life, and now he's relocating out to the country. Maybe along the way, he sees this beautiful field of wildflowers and he wants to stop, right? So his first visit, he's enjoying being out in this field of wildflowers, right? Breathe the fresh air, and he's fine, right? And then he gets in his car and he continues on his way. So the first exposure to pollen, he breathes in lots of these different pollen grains. These pollen grains, right, get lodged in the mucus, the thin mucus that protects his mucosal layer. As a result, because a lot of the pollen is made up of proteins, it kind of breaks down into individual teeny little dots. Each little dot represents a potential antigen, right? And because it breaks down so easily in the mucus, right, it's absorbed into the tissue, and you have professional antigen presenting cells like macrophages or dendritic cells, right, that pick them up. And they present them on MHC2, right? And as a result, you begin the opportunity to start selecting for, let's say, lymphocytes like T cells to begin to recognize, hey, this is something different, right? This is something we have not seen before growing up in the big city, right? And what may happen then is that you have helper T cells that activate, and they begin to help the selection of naive B cells that by chance have the right immunoglobulin that recognize the red dot, right? And so now, this is now over the course of several weeks, right? The man never reacted to this field of wildflowers on that first visit, but over time, what has happened? He's built up, right, levels of memory IgEs, and those memory IgEs coat the surface of things like mast cells. Mast cells begin to be suited up with them, and these mast cells are hanging out in his mucosal layer of his respiratory tract. Then maybe several months later, he's driving back to the city, right, to go get something. He sees the field of wildflowers again. He's like, I remember this place. I want to stop again and go out, right? But this time when he goes out in the field of wildflowers, what happens? He breathes in the pollen, and it triggers the mast cells to release their histamines, right? He starts sneezing, he starts sniffling, he has runny, itchy eyes, right, etc., runny nose, okay? So a mistake was made here, right? Because really the adaptive immune system, why does it care that there's pollen, right? The pollen's not a real threat, but somehow a mistake was made, right? And it treated it as though it was. So here are some features of allergens that actually make them very sort of likely candidates for developing allergic responses against, right? So molecular type, most of them are proteins because it's peptide fragments that are ultimately going to wake up certain CD4 helper cells, right? Many times it's low doses, they're low molecular mass, so they travel very easily and are dissolved through the mucus layer. Highly soluble, again, so they break down in the mucus layer in our body. They're highly stable, right? The more stable they are, the more extended opportunity there is for your immune system to have a look at them. And they contain peptides that are presented very well on MHC2. Right? This one right here is interesting. Function. Notice this. Many allergens are proteases. right? I wouldn't have guessed that, right? I mean, the other things like being easily soluble in water or mucus, or being lightweight so they can travel through the air, or being stable, those you can kind of guess, right? But the fact that many allergens are proteases is kind of curious. That's because many allergens resemble certain types of antigens that are presented by parasites. And these types of antigens that are presented by parasites themselves oftentimes are proteases that are made by the parasite. Remember, what is a protease? A protease is a protein that functions as an enzyme to digest or destroy other proteins, right? So why would parasitic organisms like this worm make different types of proteases? Well, they do it because in order for them to make their home in your tissues of your body that they're invading, right, they want to, right, digest your tissues to make more room for themselves, to be able to release certain nutrients that are in your tissues, and also as a defense, right? Because if they're secreting different enzymatic proteases, these proteases might interfere with things like complement proteins or the receptors on your immune cells, which are, of course, proteins, right? So as a result, many allergies happen because our immune system mistakenly sees an allergen as looking very much like a type of protease that we evolved to sort of be sensitive to, right? Let's look at this here. Here's a blood fluke, right? Big multicellular worm. Cathepsin is one of these proteases that's made by this blood fluke to kind of digest away your tissues as it's, invading your body, right? This is a papaya, right? You're like, well, what's that? It's a papaya. Yeah, you all know what a papaya is, but a papaya produces a protease, papain, right? Named after a papaya, right? Papain, the structure of papain, look at this protein. It is not a million miles away from the cathepsin that a parasitic blood fluke makes, right? And so you might say, well, wait a minute. Why does a papaya need to make this particular protease? Right? It has to do with how fruit ripen. Right? You ever think about it when you have a green, unripe fruit, whether it's an apple or a banana or a papaya, right? How green and tough and not very sweet it is inside, right? It's hard. But as it ripens, right, changes happen to the tissue of the fruit. It becomes sweeter, it becomes softer. That's because the fruit itself is producing proteases like papain that digest away and remodify or remodel the tissue, making it softer, for example. Right? So during allergies, what happens is exposure to certain types of proteins like papain from papayas or other fruit. At some point along the way, it's possible that your immune system makes a mistake and thinks that this actually is a threat, because our immune systems over hundreds of millions of years of evolution have become more sensitive to being able to pick up on things like these proteases coming from parasites as being an indication of there being a threat. Right? So that's why many types of food allergies, especially allergies to fruits, right, are this type of mistaken identity. This table down here gives you some examples, right? Different types of worms, right? They make different types of proteases that are very similar in structure to things like cat dander from cat fur or grass pollen, right? Et cetera, et cetera. Right? So interesting to think about. Right? This does not mean that if you eat a papaya, you're automatically setting yourself up for allergy, right? Because remember, we learned, normally when you're eating foods, we evolve to kind of ignore the fact that there are all kinds of weird antigens in there, right? Because otherwise we would not be able to eat different types of foods if our body always freaked out at all the foreign antigens that are in there, right? We say that normally there has to be some sort of real infection going on associated with eating something that triggers your immune system to act. Right? So it's quite possible, though, right, that at some point along the way, right, maybe you did get an infection somewhere in your body that stimulated your immune system, right? And maybe that happened to be at the same time when your body was also seeing certain types of antigens from something like the food that you ate, right? And your B cells, T cells, they don't discriminate, right? They bind whatever they bind. So it's possible that a mistake can be made. right? And your immune system then begins to learn, oh, this B cell, because it picks up on this papain from the papaya, and we're getting confused messages here because we think there is an infection going on, right? Because there is somewhere else in your body, right? You begin to select for B cells and T cells that recognize this component of the papaya. All right, so that brings me back. Hey, I finally get to finish my story here that I started before spring break. Most of you have probably forgotten my story, but some of you have asked me about it. Remember, to recap, my story was when I was a postdoc in Boston at Harvard, I used to hang out at the Starbucks a lot. And then one day I ordered this soy macchiato drink, and I had a pretty severe, probably the most severe type 1 hypersensitivity reaction that I can remember, right? My throat really swelled up. I had a hard time breathing. Even my eyelids were all puffy. And I had to go back up to my boss's office and tell him I thought I was dying, right? Unfortunately, after 20 minutes, it sort of subsided. Okay, so the rest of my story, all right. So being the scientist type that I was, right, I tended to find this particular episode far more interesting than the actual project that I was working on in the lab. So I wanted to investigate what might have happened. All right, so I went down to the Starbucks, and I noticed, right, this is a very busy Starbucks. Because everybody in the Harvard Longwood medical area around Harvard and the hospitals would come to this Starbucks. And so they were so busy that I noticed that oftentimes their sink, right, was just filled with empty sort of like cups, carafes, things like that, like the things that you use for the blenders, things like that, were just thrown in there. And there was just a lot of water in there and lots of suds, right? And I noticed that sometimes, right, they were running out of stuff. So somebody would just, one of the baristas would just grab one of the things out of the sudsy water, kind of shake it off, wipe it off, and just go ahead and use it, right? So I asked them, what's going on here, right? Could I actually take a look at the type of detergent that they were using? Because I thought maybe it's the detergent, right? And this is what I found, right? So I asked them, and they actually showed me the bottle here. Right. And this being, you know, a pretty green type of, you know, restaurant, right? Starbucks, I think, prides itself on being earth friendly and green, right? It turns out that the detergents that they used were plant-based. Right? So it says, so I don't remember the exact brand it was, but when I looked at the label to see what this is made of, what did I find? Right? That it comes from naturally derived protease enzymes. Okay. And so you might say, why does dishwashing detergent need protease enzymes? Well, it's because, right, when you have food and other crusty bits of stuff on the dish, a lot of those are proteins, right? So you want to have stuff that help break down the proteins. Right? So I was convinced then that I realized, yes, that's what it probably was, right? After many months of visiting the Starbucks, probably at some point, my immune system had mistakenly identified one of these protease enzymes that was part of the unwashed, you know, carafe or whatever that they used to make my drink that day, probably the soy caramel macchiato, right? And that caused my system to have a strong secondary immune response, right? That means at some point in the prior months, right, was when my body had made the mistake. Even though I was drinking these every day, at one day it probably started making that mistake, and it took several weeks to build up enough IgEs so that finally on this particular fateful day, when I drank, you know, the coffee from the dirty carafe or whatever, right, that this protease enzyme, right, caused my IgEs to go ahead and respond, right? My immune system thought it was doing the right thing. It thought that it was protecting me against this pathogenic threat, when in reality, there is no pathogenic threat, right? But because my immune system, right, was very sensitized to it, right, it actually was a very distressing physiological reaction. Okay. So predisposition to allergies passed on by mother to child can happen, because remember, mom's IgE, right, even though it's not passed to unborn baby in the womb, right, there is some in the amniotic fluid, and there's also some in the breast milk. Okay, so normally this is what Mother Nature intended. So for example, when baby is nursing or breastfeeding, this is another way, for mom to be able to share her library of immune memory in the form of immunoglobulins, immunoglobulin G, immunoglobulin A, and immunoglobulin E with her infant child to protect her baby, right, during that very vulnerable time when baby has yet to develop her own immune system. Right? So what may happen is that if you end up with a situation where IgEs are picking up on certain antigens that might actually be allergens, right? So if mom was allergic to something, maybe she has these improper memory IgEs that get passed on to baby, right? In baby, they may end up actually encouraging things like baby's own selection for his or her own set of memory cells like T cells and B cells to continue or perpetuate that, response. So you might say, well, what about, does this mean that this child now will always have an allergy against whatever mom was allergic to? No, right? Because you know that from your own experience. Your mom and dad might be allergic to something that you are not, right? Well, I guess dad doesn't really fit into the picture here, mainly mom, because we're talking about the IGs that mom gave you, right? But we know we can outgrow allergies, right? And I'll talk about that in a couple slides. So because many of these systemic allergic reactions can be life-threatening, it's really important, right, that we have medications on hand. So many of these are things like epinephrine injectors. So adrenaline, which is epinephrine, right, is one way to help stimulate the heart muscle to keep pumping. It relaxes airways and reduces fluid loss from leaky blood vessels. Right. So I think you may carry one, maybe you know somebody who carries an EpiPen in case they have a severe allergic reaction. Right. What's really sad as part of this whole hygiene hypothesis and more kids having allergies is now they have EpiPen Junior. Right. It's kind of both sad and really cute at the same time. Right. Okay. So asthma. Asthma is also linked to our immune system, and asthma, not surprisingly, is also linked to IgE stimulation. So when you have an acute asthma attack, that's when a specific allergen acts as a trigger to trigger your mast cells to be released. Chronic asthma, right, is oftentimes harder to deal with because with an acute attack, you can have something like an inhaler, right, that counteracts this immune triggering response. Chronic asthma happens when helper T cells continue to kind of encourage the immune system to perpetuate or select for these IgEs to stick around. Right? So if you see here in a chronic response, you will have these different IgEs that are binding to allergens. Right? They are degranulating as part of the cytokine signals that are released. This actually stimulates things like Th2 helper cells to get activated. And those Th2 helper cells, they think they're doing the right thing by encouraging the reinforcement of this immune memory, right? But in reality, in this case, the immune memory is a mistake in the first place, right? And so that's how you end up with chronic asthma. That's kind of hard to, to break free from. Okay, so in summary, there are three main ways that we've learned to kind of treat or deal with allergies. Number one is sort of the obvious ones, right? For certain types of allergies, if you know you're allergic to something, you just avoid it, right? I was making fun of that guy in one of the photos of standing in the middle of a field of wildflowers, sneezing, right? And I kept saying like, well, you're sort of dumb, right? If you know you're allergic to it, don't go hanging out in wildflowers. If he just stays away from that field of wildflowers, right, he's not going to suffer as much. Avoiding certain foods, right? That's why, all joking aside, that's why people who have severe allergies to things like peanuts, right? You have to be really careful and stay away from those things. And also cleaning the house to rid things like of dust and pollen, both of which can be triggers for certain allergies. Pharmacologically, there are certain drugs that can be used to reduce the impact of allergen exposure, right? So over-the-counter ones like antihistamines, certainly that's probably what we're all most familiar with, right? If you have a sneezing fit or something like that, right? You might, I need to take an allergy med, right? So you take your Allegra or your Zyrtec. Also, we learned about anti-IgE antibody treatment for people who have such severe allergies that it really gets in the way of them leading a normal life, right? They can undergo infusion with antibodies that are meant to kind of bind up their IgEs and prevent their IgEs from stimulating their mast cells. And the third one is immunological. This is interesting. This has to do with retraining the immune system to be desensitized to allergen. Right? This has to do with giving a patient tiny little doses of allergen, right, that they are sensitive to, and over time increasing that dosage. Why? Because our immune system is constantly recalibrating itself. Right? If you move from one location to another location, different country, different city, right, your immune system sort of readjusts to new exposures. Or forget about even moving cities from one apartment, right, in Honolulu to another apartment. Right? Maybe in your previous apartment, there was a lot of black, yucky mold in the air conditioning vents, right? And that was making you kind of sick. If you move to a new apartment that doesn't have that anymore, over time, your immune system will recalibrate, right? Because it learns, hey, right, there's no more of this happening, right? We can forget about it. Or, if you're exposed to something repeatedly, but it never really causes you to have a big response, your immune system learns to be okay and tolerate it. When I was a little kid, growing up in Boston, I actually was allergic to lobster, right? Which is kind of sad because Boston has some of the best lobster. So when I was a kid, I'd eat lobster and I'd puff up, right? So my parents said, oh, right, you can't eat lobster. But many years later, right, I found out that I was fine. Why? Because in the intervening years, when I guess I stayed away from lobster or whatever, my immune system just said, well, all right, whatever, right? We're just going to forget about this because we don't have the constant reminder of it. And so as a result, the mistake was forgotten, right? And these days, I happily eat lobster. I love lobster, although too bad I can't afford it, right? Because when I was a kid, lobster was cheap in Boston. Now it's like super expensive, right? But in the clinic, you can do this as well. Patients will be given small doses of allergen, right, to kind of increase their immune system's tolerance of it and teach their immune system to stop freaking out about it until hopefully they reach a point many months later of this constant treatment where now they're no longer sensitive to that allergen in their environment. Okay. All right, let's go ahead and stop there.